Morning, everybody. It's my pleasure to welcome you to the revamped Lipid Research Division Seminar Series. First off, I want to give a big thank you to ASBMB, who are now formally hosting this. I want to give a special big thank you to Lori Relesco, who's the one who's actually helping us manage this, along with Roya Jessup from ASBMB. And then also a very big thank you to the managers of the Lipid Research Division, particularly Rob Stylen and George Carmen, who are the ones who kind of given us this support. So for people who were at the seminar series this summer, me and Mike are still involved in running it. So that's Mike Arola, he's on here. And we have we have 4 new members of the organizing committee. And I'm just gonna introduce them here very quickly. We have Julie Brill from Canada. We have Raymond Blind from Vanderbilt in Tennessee. We have Amy Kiger from UCSD, and we have Rashid Abdou Thiem from France. So we're going to be kind of rotating through and organizing these talks. So kind of every 1 out of 5 weeks will be by a different organizer. Just on quick notes on how this session will run is essentially we are going to have a question and answer period at the end of the talk. So it's a 20-minute talk. And then people are going to have an opportunity to ask questions to the speaker at the end of the session. The way that works is there's a chat box on the right. Please write your questions into the chat box. And when we get to that question and answer session, I will call upon you. And at that point, you'll have the ability to unmute yourselves and ask the question to the speaker directly. If we don't have time for your question, we're going to have an opportunity for the speaker to write back to you in the chat box publicly and answer your question. I think that is all the kind of big details we need. And so we have 2 really exciting talks today. And so we're going to start with introducing Christiane Berger Schapitzel. I might have said that incorrectly. And she's going to introduce our first speaker. Thank you.
Hello, it's my pleasure to introduce Christine Toelzer. Christine studied biology and physics in Cologne in Germany. She also did her PhD there using mostly X-ray crystallography. Since 2017, Christine is in our lab in Bristol. So she's a postdoc at University of Bristol, and started using cryo-electron microscopy in our lab, and usually she works on transcription factors. But what happened in March 2020 is that in UK there was a general lockdown and we were only allowed to come back to the laboratory if we would perform research on SARS-CoV-2 virus. So Imre Berger's lab started to use insect cell expression and to produce the spike glycoprotein of SARS-CoV-2. And we provided this protein for serology, so for antibody testing. And we had a number of spike proteins and virions and used them for cryo-electron microscopy. Christine analyzed the structure and built the model. And during model building, she discovered an extra density, which is non-protein density in the structure of the SARS-CoV-2 spike protein trimer. And she's going to tell you more about this in her talk.
Thank you, Christiane, and thank you for to the organizers for inviting me. And I will share my screen. You should be here, okay. And today I will give a talk about this fatty acid binding pocket, what I found in the SARS-CoV-2 spike protein. And just a very, very short introduction to SARS-CoV-2, because you probably, well, everybody knows SARS-CoV-2. Yesterday I looked at the Worldometer and I found already 27 million cases around the world, and SARS-CoV-2 is affecting the world so much that some countries even had to go to lockdown. But one very short introduction to the replication cycle of SARS-CoV-2. So the virus, when it arrives to a cell, this green dots here, these are spike proteins, and the spike protein of the virus can bind to the cellular receptor. And in the case of SARS-CoV-2, this is normally the human ACE2 receptor. Then the virus can enter the cell and replicate, and afterwards form virions, which you will hear in the next talk. And more virus can be released out of the cells. So this very first step of binding to the cellular receptor is very important, and for this we were interested in this spike protein. And we started to do cryo-EM, so cryo-electron microscopy of the spike protein. What we found there, so we found two different conformation in our cryo-EM structures. And we found the open conformation, what you can see here, with about 30% of the particles, and we could resolve the structure to about 3.5 angstrom. And what you can see here, this is one of the receptor binding domains, which would bind then to the ACE2 receptor in the up conformation. So the open conformation would be the conformation which binds to this ACE2 receptor. And we had 70% of the particles in a closed conformation, and we could resolve this to about 3 angstrom. And if we apply symmetry, we even could go to 2.9 angstrom. And yeah, so in the closed conformation, the RBD is in the down conformation, what you see here. And yeah, very unexpected, we found that there was an additional density in the structure, and which was not from the protein, and which was yeah, very new and very surprising. And yeah, so we started to investigate this part. And here again, you see the closed conformation with the RBDs here, and the orange dots are where the density of this new density is. And here you see the close-ups, and we looked at all RBDs. So the spike protein is a homotrimer. And so it has 3 receptor binding domains, which you can see here, and from the top view here, here, and here. And if you look at the pockets, we could find density in all of the 3 pockets, in this blue one, in the red one, and as well in the green one. And here we have a picture of the density with the surrounding residues. That you can see that it is well defined and was nicely but empty, and we didn't know what it is. I already put the linoleic acid in here because that's what we found out, that we have linoleic acid in there. And how did we do this? So when we saw the density, so we saw the bending and the size, and from the bending and the size, we thought, oh, okay, yeah, that could be some fatty acid, and it might be that it is linoleic acid. And we looked at the PDB at other structures with linoleic acid bound, and for example, the Streptococcus pneumoniae fatty acid kinase B3, and the odorant binding protein OBP22. And you can see both of them have the linoleic acid bound, and it is bending in a similar way like we have it in our structure as well. And what we also found is that in all of these structures, there were an arginine in the vicinity of the carboxyl group, and like this, stabilizing the carboxyl group. And we have an arginine in our structure as well, the arginine 408. This arginine is coming from a different RBD, so it's a bipartite pocket. But like in the other structures, we have this stabilization. So we already thought it is linoleic acid, but we cannot be really sure from a structure which is only 2.9 angstrom, and it could be something else as well. So we did some mass spec with our collaboration partners. And what you can see here is when we put it over a C4 column, then afterwards we used this peak and got some signal in the isotope, which corresponds to 279 gram per mole, and this fits perfectly with linoleic acid, which would be 280 gram per moles. And yeah, what's so special about linoleic acid? So linoleic acid is an essential free fatty acid, and the metabolic pathway is central to inflammation and immune modulation. And these are all key symptoms of COVID-19. And also it is essential for maintaining membrane fluidity and surface tension in lungs. And the alteration of this pathway lipid composition is observed in respiratory distress syndrome and severe pneumonia. And again, both are key elements of the COVID-19 pathology. And also we found a recent publication where you could see that lipid levels are decreased in COVID-19 patients there. So yeah, we started to look closer at this pocket, and yeah, we started to look at the surrounding of this fatty acid, of the linoleic acid. And as you can see in this picture, it is fitting into a hydrophobic pocket. It's like really, the hydrocarbon tail is like really nicely fitting in this very hydrophobic pocket. And from the other side, you have the arginine and the glutamine stabilizing the carboxyl group. And here you can see an electrostatic surface presentation. In white, again, this is hydrophobic. And in blue here, you would see the positive charge, which can stabilize the head group. And yeah, I already told you now something about the hydrophobic pocket, but there are even more differences between the APO form, which would be no linoleic acid bound, and the holo form, which is with the linoleic acid bound. And as you can see here on the left side, there are some tyrosines, the tyrosine 369 and the 365, which swing open. And compared to the non-bound structure, so the gray structure here is the one without linoleic acid, and the blue one is the one is our structure with the linoleic acid bound. So it swings open and gives space for the linoleic acid. You can even see it a little bit better if we move it, if we rotate it with 90 degrees. And you can see here the gray helix, it would clash with linoleic acid if it's without the linoleic acid. It's like in a kind of closed pocket, and with the linoleic acid bound, it swings open and gives space for the fatty acid. And as I already mentioned a bit, there is the arginine 408 and a glutamine in the vicinity of the head group. So this will stabilize the head group of the linoleic acid. And this is compared to the structure without the linoleic acid bound. This is moving about nearly 6 angstrom, and closer to the linoleic acid, and like this also closer, one RBD closer to the next RBD. So if we look at all 3 RBDs, what you can see here, because there are in total 3 linoleic acids bound, and all of the RBDs are moving to each other when the linoleic acid binds. So if you compare it with the unbound form, the holo form, the APO form, then you have this bit more space between the receptor binding domains. And when the fatty acids, the linoleic acid is binding, then this gets more compact. So to remind you, so the open form is the one which binds to the ACE2 receptor, and the linoleic acid is in the closed form. So the closed form is the one which is not binding to the receptor, and for this it's not infectious. And we found that we have 70% of our spike protein in the closed form, so the non-infectious form, and 30% of the spike protein in the open form only. And yeah, the normal, the upper form had 75% in the open form and 25% in the closed form. So we started to get a bit, yeah, well, it's exciting that it changes, but also we use this spike for serology assays. So we had to make sure that it's still binding to ACE2. And yeah, for this, we looked at the interaction of our spike protein with ACE2. And here you see again the picture of the receptor binding domains. And in red, you can see here the receptor binding motif. It's not directly, yeah, it's not very close to the linoleic acid, but as everything is, as it shifts the equilibrium to the closed form, we had to check that it is still binding to ACE2. And yeah, you can see here a size exclusion chromatography. In yellow, this is a ACE2 alone. In green, this is a spike protein. And when we mix them together, they shift. And you see on the STS page that this peak number 1 is really spike and ACE2. So we could confirm with it that it's still binding. Then we also did some competition ELISA, and this works like this, that we immobilize ACE2 on a plate. And then we have RBD HRP, which would then bind to the ACE2. And we titrate our spike in there, and then the spike would compete with the RBD and would then lower the signal. And as you see, with higher concentrations of our spike protein, also we get less signal. So this also shows that it's still binding to ACE2. To do a little bit more sensitive method, we also did SPR, and the binding constants were more or less in the same range for the APO spike and for the LA bound spike, but we could see that the response every time went lower for the LR bound spike compared to the APO spike, which would make sense because for the LR bound spike we think that the closed conformation is a more prominent one, so it cannot bind as much as for the APO spike. Then we got very excited and thought, okay, maybe this is also conserved, and maybe we find this in other coronaviruses as well. And we looked at the sequence, and it is completely conserved for the SARS-CoV-1. And here's a picture of it. So, well, with a sequence comparison, you see it's really completely conserved. The whole pocket, together with the polar lid, is completely conserved. And if you look at the structure, you can see that the purple one, the SARS-CoV-2, the SARS-CoV-1, looks exactly like the SARS-CoV-2, but in the not locked form. And it looks like it can move as well, and it could, yeah, as well bind the linoleic acid. For the MERS, we saw, well, it's not as conserved for the MERS as for the SARS-CoV-1, but we found other residues which could maybe facilitate the binding, and we found this lysine 502, and the hydrophobic residues in the pocket were normally only replaced with other hydrophobic residues. So it would be really nice to find something what we can fit in this linoleic acid pocket, and if it's also the same for SARS-1 and MERS, that would be an antiviral which you can use for not only for the SARS-CoV-2. And oh, that was the wrong direction. And So this is the idea what we have now. So without linoleic acid bound, we see the infectious form is favored, and the open form is about 75%. And already with the linoleic acid, we shift this equilibrium, and we have 70% in the closed form. And if we now can find a small molecule which binds into the pocket and then locks the closed conformation down, irreversible, like kind of a super linoleic acid antiviral, we can increase the non-infectious form to about 100%, and like this, then decrease SARS-CoV-2 infectivity. That this is possible, there are examples, so I will not go into detail in this because of the time, but there are examples that you can exploit a hydrophobic pocket and then abolish the receptor binding. And this is one example, the Wnt compounds, which bind to a hydrophobic pocket in the rhinovirus and can displace the fatty acid and then hinder the receptor binding. And these are already successful in the clinic phases. Okay, so to summarize everything, we found a linoleic acid binding pocket in the SARS-CoV-2 spike, and this is the first direct structural link between linoleic acid, COVID-19 pathology, and SARS-CoV-2 virus itself. And this is a bipartite pocket, so it connects two RBDs within the spike trimer. And it stabilizes the locked form of the spike with the receptor binding domain locked down and tucked in. And the closed form is a non-infectious form. And this pocket appears to be present in other severe causing coronavirus, disease-causing coronaviruses. And like this, it's a very good druggable pocket and a good target for new antivirals. And yeah, with this, I like to thank the Berger and Schafitzer group, especially Imme and Christiane, because this project wouldn't have been possible without them. And also Kapil, Satish, and Ufuk, who did a lot of the work. And yeah, we had cooperation partners in Switzerland and in Germany as well. And yeah, thank you for the funding as well, and thank you for all to all of you for your attention. Thank you. And I'm happy to take questions.
All right, thank you, Christine. That was a great talk. All right, so I'm going to be calling on people here. We've got a lot of questions coming in thick and fast. I don't think we have time to get to all of these. But the first question is from Andres Arango. Andres, you have the ability to unmute yourself now.
Great talk. So I'm curious, did you see or did you look into the effects of other fatty acids such as arachidonic acid? I think other people asked this question too.
Yeah, well, in the mass spec, we saw that it is fitting to the linoleic acid. So with the SPR, we first already only did the linoleic acid, but yeah, we were thinking about other fatty acids as well. But at the moment, we only looked at the linoleic acid because everything fits very well, and yeah, that's mainly the time reason as well.
All right, next question is from Miguel Guion.
Oh, hello. Actually, my question was very similar to that of Andres, but I have another one that's related. And first of all, let me tell you, it was a very nice talk. Thank you. But it looks from your images, the fatty acid goes tail end first. Did you ever check if it would also take, say, a lysophospholipid or a phospholipid that has linoleic esterified?
Yeah, so we didn't test it yet. With bigger compounds, it would be a bit difficult in the pocket actually, and yeah, well, we haven't tested it, but from the space, if we go bigger, it would be difficult to fit. But well, everything is quite flexible in this part, so there's no reason that it cannot adjust a bit. Yeah, so at the moment it's more fitting to the linoleic acid than bigger compounds.
Okay, thank you.
Okay, the next question is from Shashank Pant.
Yeah, hi. Very interesting talk. So I'm curious about, is LA binding cooperative in nature? Like the binding of the fatty acid, once it docks itself into the interface of the two RBDs, does it help or facilitate the binding of the second fatty acid?
Yeah, that's a really good question, and I really would like to know this as well. At the moment, we don't really know. So with SPR, so we started doing SPR for the RBD, for the linoleic acid binding as well. But we can only do it to the receptor binding domain alone, so not to the full spike protein. Because with a full spike protein, it would be just a big protein and a very small linoleic acid, so you will not see the change. But and with the RBD, you cannot really see the cooperativity. And to say this from the structure, that we cannot really answer from the structure. So we really have to test it with other assays, and well, this is ongoing work, but it will be a bit tricky because we have to do it on the spike protein and not on the receptor binding domain alone. And yeah,
and just to follow up one.
Yeah.
So, yeah, thank you.
Maybe it's
So
in the
just
interest of time, Shashank, no follow-ups, just because we've got a lot of questions here waiting and a limited period. All right, Ray, you are up. One question, Ray.
Yeah, so I'm wondering how dynamic you think there might be kind of exchange between this and what the expression system was.
Yeah, so the expression system was insect cells, but there are also now structures out which have the density as well. Maybe I can show you. So there are other structures which are expressed in mammalian cells, so it's not an artifact of insect cells. And so that was one question. And how dynamic? Yeah, this is also a bit hard to say because when we freeze it, we only see like a snapshot of it. But it will be dynamic, because if we don't have this dynamic, the change in open and close conformation, we would not see a binding with ACE2. So it's definitely dynamic. How dynamic we cannot say, and we also don't know if we really have it in the open conformation because we cannot see it with our resolution. It's a bit the resolution is not good enough for the open conformation, but we should have it because we track it along the whole purification. So we probably picked it up in the insect cell expression media and then purified the protein, and it still holds on to it. So the linoleic acid might go maybe a little bit out, but it's not going out completely. And we only have the dynamic with the open and closed conformation, but still have the linoleic acid in there.
All right, next question is from Fran Barrera.
Hi, this is exciting research. So if there's hope for linoleic acid becoming an inhibitor of the virus, you would like a high affinity to the receptor binding domain. Have you determined this affinity?
Yeah, so we haven't determined the affinity to the full spike protein because this is not possible with SPR, so we have to see. But to the receptor binding domain, we could find a binding constant of also low nanomolar range, so 41 nanomolar, and it will be probably better for the spike because we don't have this bipartite pocket. We just have only one receptor binding domain, so the KD will be better for the spike protein then.
That's wonderful. Thank you.
All right, probably in the interest of time, we're gonna have to end the questions here. Apologies to everyone. I see George Carmen is next. So George, one of the managers of Lipid Research Division, I apologize, but you know, in the interest of time, we'll move on. So as a reminder, Christine, if you could please answer the additional questions in the chat box publicly, so everyone can see the answers to those. And with that, I will hand over to the next speaker. And I'm going to hand over to Dr. John Briggs, who will introduce our next speaker.
Hi, so the next speaker is Zunlong, who's a postdoc in my lab at the MRC Laboratory of Molecular Biology in Cambridge. Zunlong did his bachelor's in Wuhan in China, which has obviously been in the news a lot recently related to SARS. And Zunlong had moved to Georgia Tech in the US to do a postdoc with Liz Wright, where he'd started doing cryo-electron tomography of viruses, especially measles virus. And after finishing his PhD, he moved to my lab, where he continued to do cryotomography and cryo-EM of viruses. He started working on the filoviruses, so Ebola, and on HIV, and also a little bit on influenza. But he became very interested in working on SARS early in the pandemic, and he's going to talk to you about the work that he's been doing in the lab together with a couple of the other members of my lab. So Zunlong, over to you.
Okay, thank you, John, and thanks to ASBMB for the invitation. So today, I would like to share the stories we have on SARS coronavirus, especially the spike protein on the virions. So this is a droplet. And then if we think about the process, well, this is from David Goodsell, like the droplets, the coronavirus is, inside the droplet, and then it's transmitted through that. And it's really small, and then we don't know, well, so it's really small, and I would like to study that, especially like in the context of the full virion. At the beginning of the pandemic, there were first images of first micrographs of the coronavirus, that time was called the novel or new coronavirus 2019. On the left image, as you can see, maybe I'll just change it to this laser pointer. Here we go. So on the left image is a negative stain image of the, you can see two coronavirus with the crowns surrounding by it. And on the right is a thin section of the airway epithelial cells. You can see the coronavirus is here, and then the cilia, as well as the viral factories inside with the coronavirus, SARS coronavirus in it. Now I just get back to a bit about the background for the SARS-2 coronavirus. It's a huge RNA virus. It has a huge RNA virus genome, has 30 kilobases. It's the largest, actually, RNA virus genome. It's positive sense, single-stranded. So now this virus has a protein S. It's on this virus surface here. And then inside it has the membrane protein, or you can call it a matrix protein, embedded in the membrane. And then inside the lipid bilayer has the nuclear protein that has the RNA encapsulated in it. So on the left side is 2 micrographs of the coronavirus tomographic slices. As you can see, the crown of the coronavirus, the spikes protruding from the lipid bilayer. So I'll get a bit more details later as we go on to the talk. So at the very beginning of this year, in February or March, there are 2 groups solved the spike protein by single particle cryo-EM. As you can see from Jason McLean's group and David Whistler's group. So what they found was that, I mean, it's amazing that, they solved it. This study was done like in actually 12 to 13 days, which is, I think is really impressive. What they saw was that the coronavirus resembles a pre-fusion state. They both of them have seen that, and also they have open conformation. One of the RBDs comes up, and then closed conformation that all the three RBD or receptor binding domains is closed in those states. Now, if we think about the spike structure on coronavirus, what they did, I think, is really good. But I feel like the need of the spike structure on the virions is not characterized. So I will just show this one here. So this is a tomography of the purified spike proteins. What you see is that those purified spike proteins in the thin layer of ice, and we can get the three-dimensional structure of the spike proteins. However, by doing so, we are losing the virion context. For example, we do not know whether spikes are clustering or not. We don't know their relative location. And if we show this one here, this is the structure solved from Jason McLellan's group, that you can see the spike is quite flexible in both the receptor binding domain as well as the membrane proximal domain at the stalk region here. So we don't have that by doing single particle cryo-EM. Now, to do that, our group collaborate with groups in Heidelberg University that we wanted to see if we can solve the spike structure on the virions. So this is one of the collaborators, Marco Curtis. He's carrying a inactivated infected cells, infected with SARS coronavirus 2. So here is the image of the cells cultured on the grids. That was one of our first avenues that we took to solve the structure. So now, I just talked a bit about the sample preparation that we did. So first we infect the cells at MOI at 0.5. And then after 2 days of infection, oh, by the way, all this sample preparation is done in Heidelberg in our collaborator. And we harvest the virus 2 days post-infection, and then we fix it in formaldehyde. So after that, we have 2 ways to collect the virions. The first one is we directly take the supernatant of the infected cells. We do a clarification by a low speed spinning, and we get rid of the cell debris, and then we have the very diluted supernatant of the cell. We call that unconcentrated sample. And then we also have the concentrated sample by purifying the sample using 20% sucrose exclusion. So if you compare this to method, the supernatant method is really nice because we don't need to go through this. We don't have to let the sample go through high-speed centrifugation, which can potentially damage the structure of the virion as well as the structural components, for example, spike. But if we go through the high-speed sucrose exclusion, then we can, of course, concentrate the virus particles, and that might be able to help us do high-resolution structure determination using cryo-EM. First, we characterize the spike. Hold on, sorry. I'm sorry, this is really strange. Oh God. Okay, I cannot move to the next slide if I will just to go here. Yeah, here we go. Oh, I'm sorry. Screen automatic. Here we go. Okay, so on the right side of the image, I apologize for that. So the spike protein is has the S1 part and S2 part here shown. So the S1 so we used antibody. First, we characterized the virions in both Viro E6 cells and the Calu3 cells. As you can see, in the virions, the spike has the S0, which is the whole S, and then S2, which is just this part here after cleavage. That you can see in both of the samples, they all have the spikes cleaved into the S1, S2 portion. So I need to mention that the antibody we used is for the C-terminal region, so that we don't see a S1 band in here. And you can see, also you can see that the portion of the S2 to the S0 is a bit different in two different cell lines. Now, I just go to the tomography part. First, we image the supernatant. The supernatant, as you can see, they are really low in various numbers. The first image, the left image you can see is just one tiny virus particles in the big field of view under the electron microscope. And on the right is a tomographic slice of that virus. By imaging this sample, we have a hard time to locate or find find rhythmal regions to collect the data of this one. And then I just show you a tomogram, or actually on the left is a tilt series of this virus. And on the right is a tomogram. It's a tomogram, like slicing through this virus. So what you see is you can probably identify features of the virions. You can see the lipid bilayer. You can see the spikes protruding from the lipid bilayer. And if you notice carefully, you can also see the granular densities inside the viruses. I just show a couple more examples, just give an idea of the virus particles in three dimensions. So yeah, some are bigger than the others, some are a bit smaller, and some of them have a bit more densely packed spikes, and while others have maybe not so much relatively. Okay, move on. Next, we also image the concentrated sample, because that one has a potential that you can do high resolution structure determination. As you can see, this field of view has a lot more virus particles. If you recall the previous image, that has much fewer virus particles. And we characterized the morphology of the viruses. This image shows the virion diameter on the x-axis, and then this one shows the number of virions on the y-axis for both the unconcentrated sample, which is the supernatant, and the concentrated sample, which is the from sucrose cushion pellet. As you can see, in both cases, the average virion diameter is about 90 nanometers in diameter. This is measured from viral membrane to viral membrane, not including the virus spike. At the same time, the number of spikes per virion, they do vary. As you can see, some of them has very low number of spikes on the surface. And then on average, it has about 24 spikes per virion in the unconcentrated sample. So next, we just wanted to ask the question is that, what is the structure of the spikes on the virus look like, and how far we can get by analyzing the subtomograms of those. So we collected a total of 215 tomograms of that, and then each tomogram has on average about one virus particles inside, and at the end we used 156 tomograms, and there are 179 viruses total, and we picked 4,000 particles of that. So by subtomogram averaging, we get 7.7 exon structure out of the total data set. And that was C3 imposed, as you can see. So next, we want to ask the question is that, can we classify the receptor binding domains of those spike proteins? So we just use this mask here, as you can see on the top view and side view of this region. rely on to ask the questions that how many spikes have this part that's in open conformation or closed conformation or any other else conformations that we have. We found in the unconcentrated or the supernatant sample, we have 45% of the monomers because it's a C3 symmetry. So we just analyze this region here. 45% of them are in closed conformation, and then 28% of them in open conformation. As you can see, this one stands up relative to this one. And then there are 27% of them are relatively weak. But if you increase the threshold, that will be in closed conformation instead of open. We did the same for the concentrated sample or the sucrose cushion pallidus sample. There are 67 of them are in closed conformation and 33% of them in weak conformation. However, we didn't see any open conformation, which is quite interesting, which we can get that at the end as well. So by doing the classification of the RBDs, we can then deduce what the whole trimer look like, because we know each monomer is in open, closed, or weak conformation. Then we found there are 3 different classes. This one is all the RBDs in the down state or closed state, and this one in the up state. There are 55% of them. And then there are also 2 open RBD conformation as well in the spikes on the virion, which is not actually found in the single particle analysis. In a single particle analysis, there is a structure that has 2 open conformation that was published that was using the engineered mutants to have that kind of conformation. We also found majority of the virions are in pre-fusion state or in one of those 3 states. There are very few particles that are actually in the post-fusion state, about 1 spike per virion or less in the post-fusion state. So now we also noticed the spikes, they are not always like pointing straight up out of the viral membrane. Sometimes they have very high tilt angle, as you can see in this one here on the bottom right. This is the virus particle. As you can see, this spike here, this green arrow indicates the projection of the spike here. This one is highly tilted. This is about 76 degrees if you measure the angle, the normal angle of that. This is 76 degrees. So during the whole population, this is a whole population plot. So you can see the number of spikes that they tilt like basically from 0 to 90 degrees or so. And then they at 50 degrees, after 50 degrees, the amount of spikes, the number of spikes tilting above 50 degrees will decrease. And also we averaged the tremors within certain location and orientations of the spike. We found that there is a hinge at the basically the base of the head, but at the top of the stalk right here is the hinge where it starts to tilt at this region. So this hinge or this stalk region is quite flexible. It allows the spike to tilt actually up to 90 degrees, and it has a function that also was described in Martin Beck's group, that this can allow the spike can kind of rotate or rotate around the viral surface so that it can have a higher chance to bind to the host receptors ACE2. Next, we built the 3D models of the spike tremors on the SARS-CoV-2 virion. As you can see, there are 2 examples of the virions. what we can see is that, first of all, it's not highly dense, packed, like the surface is not covered with spike proteins. Number two, there's no obvious clustering, and some regions don't have any spikes, some regions have more spikes than others. Number three, there are about 1 trimer per 1,000 nanometers squared. So this number is really low if you compare to the influenza virus. Influenza virus surface spike has about 1 trimer per 100 nanometers squared. So you can see this one has much fewer particles on the virus surface. And by the way, these models are real authentic models because all those positions and orientations as well as the conformations were determined from subtotal averaging. So the last part of the talk will talk about the high resolution structure of the spike that we determined in situ on the virus surface. This is a micrograph that has the virus palatted through the sucrose cushion. As you can see, there are a lot of them on this image. So we use topaz to pick the particles on this micrograph. And then the workflow is basically that we do 3D classification. So we supply a reference and we do 3D classification of the spikes, and we get good classes, and we further classify that. And then after that, we get we further classify that, and then with the 55K particles, we obtain a 3.4 angstrom structure. Now, we ask the question is that, what about the conformations of the RBD regions in the spikes? So we did the focus 3D classification on this region here, on the RBD region. We found that there are classes of this region that are weak, like it's weak here, and then other classes that they are all closed. So we then obtain structures based on the presence or absence of the region, and we obtained two separate structures at 3.5 angstrom, which is all the RBDs are closed, and another one is the 4.1 angstrom structure that at least one RBD is weak. So now this what we see from this structure. First of all, there are 17 glycosylation sites that were observed as shown colored in orange. And out of this 22, 17 were characterized. And we have electron density of that. The other 5 were either in the NTD, the N-terminal domain that's very variable, or in the stop region that's also very variable. The other point I want to notice is that in our high resolution structure, we can clearly see, of course, the alpha helix and the beta sheets, the separation of the beta sheets, and also, more importantly, our strain that was used is a D614G mutation. So this 614 amino acid is a glycine instead of a D here. Now, in our structure, we cannot see a stop bridge in between 614G and 854K here. In other viruses, when this is a D, there is a stop bridge here. So another point I want to mention is that follow up on Christine's talk, that we didn't see any lipids that's a bond in our structure. So yeah, with that, I would like to conclude that we characterize the SARS-CoV-2 variant morphology by cryo-electron tomography. There are 2 distinct conformations of the S trimers, basically prefusion, postfusion, with the prefusion dominance, 97% of them. The RBD domain that we found, there are different percentage of the RBDs on the viral surface, with about 33 are in 3 closed, and 55 are in 1 open, and 14 in 2 open conformation. We also built the 3D models of the S trimer on intact the SARS-CoV-2 virion based on their location, distribution, and orientation. Also, very importantly, that we determined the high-resolution structure using using the in situ samples, that we directly solve the structure of the spike on the virions to 3.4 angstrom. And that might allow us to study the neutralization antibody binding in the contact of the virions instead of the purified system. And with that, I would like to thank a lot of people that's involved. Absolutely, this is a team effort. So Kino, he did a lot of the rely on classification and refinement work, and Quinn also postdoc in our group, that he did most of the single particle work. And Yosenko and Jules also, they really helped with the reliance on the topos peaking, as well as the process of the single particle process. And also, of course, Leslie from our visual aids, that he really helped with this model building of the spikes, and our collaborators from Heidelberg, especially Merkel and Wojciech, for their sample preparation. And with that, I'll take questions.
All right, Zunlong, thank you very much for a really exciting talk. I've actually got a question here that maybe I'll, seeing as I'm moderating, get the chance to ask the first one. So quick question on the altered orientations that you're seeing at kind of the membrane surface. When you showed those orientations, it looked like there were a lot that were even kind of crossing 90 degrees. They were low frequency, but there were some that were, you know, even past that point. Were you always seeing the same part of the spike kind of highly tilted? So you see that those, you know, ones over 100 degrees. Did it look like there was any part of a spike that actually seemed like it might bind the membrane, or was it kind of a random orientation across those high angle spike particles?
So thank you, John. I think that's a great question. We actually spent quite a lot of time on analyzing that. So the bottom image here, the bottom graph here, the red line actually shows that basically there is noise in the image processing that we all know. We do not believe there are spikes passing over 90 degrees that kind of, you know, goes back into the membrane, which is, you know, biologically impossible. So because the searching is, you know, 360 degrees, it can potentially go, you know, go to the other places when there's noise. So we basically cut a line here that we think this region here is, well, okay, so let me explain this figure here. So this is angular density from 0 degrees, as you can see. Basically, this number of spikes here. As you go to higher angle, you're supposed to have more spikes if they are evenly distributed, because angle is just bigger. So you just divide this number of spikes over the sine of this angle here, that you get this plot here. And then what you see here is that we think those are noise because they just don't exist. And you can see at 50 degrees the decrease in density of the spikes. I hope that answers the question.
Yep, that answers it. All right, Ray, you've got a question?
Yeah, you had mentioned in one of your slides that the size of the outer membrane of the virions, that was real, that wasn't an intended part of the thing that you were showing. So I'm curious if the S trimer concentration on the surface of the particle varies from particle to particle, and if that is actually something that you observed.
The number of spikes on the viral surface.
Yes. Is that constant, or is the concentration on the surface constant?
So this plot here is a scattered plot. As you can see, this is the virion diameter. Each point is a virus. You can see the number of spikes on each virus with different diameters on them. So the number of spikes on each virus, they are not constant, they vary.
So if you have a different number of spikes on each virus, that's the surface concentration will be different on each virus. So if you are the virions that have higher surface concentration of S trimer, are they actually more virulent? Can they infect better?
That we don't know because we need to separate those out and do the infection assay, which I think we just cannot do it. But you can see in these two viruses here, right? This at least on this side of the virus, they have fewer particles. On this side of the virus, they have more particles, as you can see. They are, I guess if you're talking about the infectivity, that might relate to the receptor binding domains conformation. As you can also see, they are pretty random. And yeah, so I'm not sure if there's a correlation, but they probably will, I guess. But we need to, I guess, answer that question somehow.
All right, Roger Williams, you have a question? Roger, you gotta unmute yourself.
Okay, I was just wondering, is the formaldehyde treatment, is that necessary to maintain intact viruses, or was that to inactivate the virus?
I'm sorry, something about the formaldehyde. Could you repeat that?
You did a formaldehyde treatment. Was that necessary to get intact viruses, or was that to inactivate the virus?
So the question is, does formaldehyde, is that to get the full virus particle, or is it just to inactivate the virus?
Yeah.
That is to inactivate the virus.
Okay, so it doesn't, if you were to do this with a full virus without the formaldehyde, you would still be able to see intact viruses.
One more time?
I'm sorry, you could still see this, the formaldehyde treatment, is it possible to do this without the formaldehyde?
Yes, you can. You can use, well, you can either study the infectious particles, with an electron microscope capable to image those, or you can use other inactive reagents.
Okay.
In the first case, I don't think we have that capability to image the infectious particles yet, because they simply cannot get out of the category 3 lab. However, if I can share with you another slide, actually, that might be interesting to you, is this one. So there are other groups using, I forgot the name, it's a nucleic acid inactive reagent that they inactivate the virus with that, and they only see the post-fusion spikes. As you can see, the needle-like, they're really long, that give different, different particles.
All right, I think we've got our final question here. Oh, we got one more coming after, so we have 2 questions left. So that's probably where we'll cut it with these 2 questions. So the next question is from Rami Amaro. You should have the ability to unmute yourself. Rami, are you there?
Unmute. There we go. Thanks, John. I just wanted to say your work is just fantastic. Just so brilliant. I had just 2 questions. My first question was, can you tell us anything about the M proteins? Are you sort of trying to figure out exactly what's going on with those? Does your data reveal anything?
M protein.
Yeah, the M protein.
You mean the nuclear protein or matrix protein?
No, the matrix protein,
Right, the matrix protein. Our data, I do not know. I mean, the model we have currently is that they are kind of buried inside the lipid bilayer, which is very different from all the matrix proteins that we have studied. But no, we don't have any yet. Or we don't know.
Yeah, we've been just trying to also just try to figure out what that looks like.
But also all the data, both the tomography data and the single particle data, they are all deposited to the Empire, so everyone has the access.
Perfect. That was my second question. All right, thank you.
Yes, both of them, all of them are deposited.
All right, the next question is from Viviana Monge-Galvin.
Yes, hi, good morning. Thank you so much for the talk. It's brilliant work. I know that you mentioned that different virions have different concentration of spike proteins, and on the image, on the sample image you showed, when you mentioned that there are no specific clusters or patterns of S proteins on the virion surface, you can sort of see some aggregates, right? And then some regions that are depleted. I don't know if you have any leads or if there any way to check via your studies whether maybe some lipid clustering is occurring at those regions, or other viral proteins that may be causing the S protein to be just on those particular regions of the virion.
Right. So I think we also would like to know the question or the problem of this study is that maybe with a much larger data set, we might be able to classify those out, but we have only 4,000 particles and 179 viruses in this data set that we can see the different conformations. I did not mention what we can get from this, but from this one, we also did tomography of those concentrated particles, and we did not reveal any open conformations, as you can see from both the high resolution structure as well as from the tomography subtonal averaging structure that we didn't resolve those. So we do have lots of particles from this data set, but those do not have the different conformation of spikes, which might be lost during the purification process. So yeah, we're a bit limited. I hope that.
Thank you. Congratulations. It's great work.
Thank you.
All right. I think with that, we've answered all the questions. Just these people can't unmute themselves. Just wanted to for everyone, congratulate the two speakers and thank them for their participation. Those were really great talks.